After initiation, we will start with the second step of translation, that is protein biosynthesis. The second step is known as elongation. Elongation is considered as the second step of translation. The term itself suggests its meaning, that is, in this step, the peptide chains formed by the amino acid molecules will enlarge by continuously adding with one another. In this, first of all, the tRNA molecules, that is transfer RNA molecules, that carry their specific amino acid molecules as said earlier that the amino acid molecule is carried by tRNA within the cytoplasm. Therefore, the transfer RNA is mainly responsible for the transfer of amino acid molecules. These are the amino acid molecules that possess the anticodon present on tRNA. Such anticodon is specific to the genetic code that is present on mRNA. That is, as said earlier in the initiation step, that in the mRNA molecule, the amino acid synthesis begins with the help of amino acid methionine, which is formed on mRNA, and correspondingly its anticodon is formed on the tRNA molecule from where the transfer of amino acid occurs. In this step of translation, that is in elongation, there are specific proteins known as elongation factors and GTP, which acts as an energy source helps in carrying out this function of elongation of protein biosynthesis. This figure depicts the elongation that occurs to form protein. As it can be seen from the figure, methionine, that is depicted by MET, at the first position is joined to the second position of amino acid through a peptide bond, that is methionine and PRO, that is proline. They both are connected with a peptide bond. Now, the tRNA molecule, as said earlier in the initiation step, that this transfer mainly occurs due to enzyme known as amino acyl tRNA synthesis, and in case of methionine, that is formyl methionine tRNA synthetase. On this basis, the formyl methionine is released, as a result of which the dipeptide between the two amino acids gets formed. And in this manner, more amino acid molecules get attached to it, and thus the elongation continues. This process repeats until reaching a stop codon. So, let us understand that how this process of elongation occurs. As said earlier, that with the help of elongation factors and GTP, this process occurs. In this, methionine, that is MET, is always occurring at the first position. That is, the protein biosynthesis always starts with the amino acid molecule methionine, and therefore it is also termed as the initiator codon. This we have already studied in the genetic code topic. Now, this methionine is attached to the second amino acid, which can be any, through a peptide bond. Now, the tRNA molecule, which is responsible for the transfer of amino acid, carries the formal methionine and releases it outside. Following it, a dipeptide gets formed. That is, through a peptide, two peptide bonds combine between the two amino acids to form a dipeptide. Following it, the ribosome that is present within the mRNA molecule moves along mRNA, which is formed at the end of transcription in a specific direction, that is 3' direction. By a specific distance, and such a distance is generally of 1 codon. That is, the space between the two is almost about a codon. As a result of this, the tRNA that was responsible for the methionine transfer is released from the surface of the ribosome. Thus, the tRNA molecule that was carrying the dipeptide bond now occupies the vacant region. Thus, this is the tRNA molecule that later occupies the vacant region. That is the region where there was nothing. In this manner, one by one, that is gradually with the increasing amino acid, tRNA molecules that carries the respective amino acids gets arranged in a sequential manner. And accordingly, the peptide chain, that is the bond that forms a chain, elongates. Thus, this elongation of peptide chain results into the formation of polypeptide. Such a phase is known as elongation phase. Thus, this was all about the second step, that is elongation. Following it comes the third step, that is termination, where the complete synthesis of protein occurs. Following it, the third and the last step of translation or the protein biosynthesis is termination. Termination, the term itself suggests that stopping anything. In this process, the genetic code that is located at the 3' end of mRNA does not exactly indicate the position of any amino acid. So, such a codon is called termination codon or nonsense codon. Therefore, on this basis, as described earlier in the main properties of genetic code, it was described that there are few codons that indicate the termination at the synthesis of protein. That is, these codons do not code for any amino acid. Such codons are known as termination or nonsense codons. These codons are of mainly three. First, UAA, second, UGA, and third, UAG. These three are only called termination or nonsense codons, and therefore the function of these codons is mainly to release the synthesized polypeptide chain from ribosome. Now, this termination or nonsense codons are very essential in protein synthesis because although protein gets completely synthesized at the second step, that is elongation, but this synthesis should be stopped at a particular region. So, in order to stop such a synthesis, termination step occurs, where with the help of any of these codons, the termination of protein synthesis occurs and the polypeptide chain gets released. In this, from the first amino acid of the released polypeptide chain, that is formyl methionine, as said that formyl methionine is the first amino acid that is formed during the peptide chain formation. So, out of this, the formyl group gets released. After which, the polypeptide chain becomes ready to perform its function. That is, although the protein chain or the polypeptide chain gets formed, but in order to activate its function, the initial amino acid group, that is formyl methionine, is necessary to release. However, there are a few times where along with formyl group, even methionine gets released. This figure depicts the termination process where, as it can be seen, that the tRNA molecule, which after getting released from the ribosome, terminates the step, as a result of which the polypeptide chain containing a long sequence of amino acid gets released. Thus, this is all about the translation process, after which ribosome containing mRNA molecule finally forms the protein